Welcome back, students, and welcome to our second lecture on chemistry, elementary introductory chemistry. This lecture, instead of talking about basic elementary chemistry, we're going to talk about the macromolecules. Macro, of course, means large, and then molecules, you know what molecules are at this point. So biological macromolecules are very large molecules that are used to put together the cells that form the human body. So what I'm going to discuss first is what exactly are macromolecules and how are they made. They're made through something called modular construction because you put together a series of the same small parts, and if you put together enough of them in slightly different combinations, you can achieve a great deal of diversity amongst really big, really complicated, really important macromolecules. And usually these macromolecules are put together by taking smaller, what are called monomers, and then you put two monomers together and you split out a water molecule, which is known as dehydration synthesis. And if you keep doing that, you can make a large molecule. You put on one monomer after another, and in order to hold it on or attach it on to the larger molecule that you're building, you remove a water molecule every time. The reverse is called hydrolysis. Hydro means water, and then lysis means to break apart. So hydrolysis means breaking apart large complicated macromolecules, breaking them apart back into their individual monomers by adding water. Okay, so then once we've learned how to build and take apart these macromolecules, we'll discuss the four major classes of macromolecules that make up every living thing. And those four classes of macromolecules are carbohydrates, which are commonly known as sugars, lipids, which are commonly known as fats and oils, proteins, they're not commonly known as anything except proteins, and nucleic acids. Nucleic acids meaning DNA and RNA. Genes, of course, are made out of DNA, and then DNA is copied temporarily into something called RNA, and genes encode proteins. So of the four classes of macromolecules, only one of them is actually encoded in our genes. The other three are created by chemical reactions that are mediated by enzymes, and enzymes, of course, are proteins. And so some of the proteins that our genes encode are enzymes, and those enzymes will mediate or control or facilitate chemical reactions that lead to the creation of the other three types. All right, so modular construction of macromolecules from simpler monomers. So if you put together, if you have one unit, whatever that unit may be, you would call it a monomer. And if you put together several monomers into a chain, you end up having a polymer, and that is called polymerization. Right? So if you put together monomers, you can create polymers. Okay, so if you ever played with these toys when you were a child, I know I did, you know these toys are called Legos. They have these little blocks that snap together. And if you take these toys, these little blocks, and you snap them together, you can snap them together in various different combinations to make really complicated structures. And that's kind of what we're doing when we build proteins out of monomers called amino acids, or if we build lipids out of monomers called fatty acids, or if we build polysaccharides out of monosaccharides. We're simply taking these simpler building blocks and putting them together one at a time to build a really complicated macromolecule. Similarly, if you have any experience with electronics, you know that if somebody gives you a box full of capacitors and resistors and transistors and connectors, and you put them together in one order, you can build a radio. If you put them together in a different order, you can make a television or you can make a computer. The components are the same, but depending on what order you put them together in, you can build different things with those simple components. Right? So you put them together in one order, you build a computer. You put together the same components in a different order, and you build a television or a radio. Okay, so that's basically what we do to build each of these four classes of macromolecules. Okay, so if we build carbohydrates, which are also known as polysaccharides, the monomer units, the small building blocks that we use to build them out of, are referred to as monosaccharides. There is a macromolecule called lipids, and in order to build lipids, which is quite a large macromolecule, we build them from the smaller building blocks that are known as fatty acids or as hydrocarbons. Proteins are large macromolecules that are put together one building block at a time, and those building blocks are referred to as amino acids. And finally, nucleic acids, the things that our genes are made out of, are built by putting together monomers called nucleotides. Nucleotides. How do we put them together? As I mentioned, you put them together by splitting out a water molecule. If you put two amino acids together and you split out a water molecule, the two amino acids will be joined together by a covalent bond. In the case of proteins, we call that bond a peptide bond, which we'll talk about in a minute. But in all cases, we're putting things together, but usually by removing a water molecule, and that is referred to as dehydration synthesis. So hydration, of course, refers to water, and dehydration means to remove water or to have water being absent. Right? By the reverse of that, the converse of that is if you want to break these macromolecules back into their individual building blocks, back into their individual monomers, you do so by adding water. So hydro refers to water, and lysis means to break apart. So hydrolysis means to break apart covalent bonds by adding water. All right, here's an example. These structures represent glucose. Right, so one of these things here is a glucose molecule. Here we have a glucose molecule, and here we have another glucose molecule. And notice that on this side of the glucose we have a hydroxyl group, right? And there's another hydroxyl group, and here's another hydroxyl group. And if you take the H from this hydroxyl group and you take the OH from that hydroxyl group, that together is H2O, or water. And if you split water out, you will have created a bond, a covalent bond between the two glucose molecules, and you will have thrown away the water. Right, so that is called dehydration synthesis. And just in case you're curious, what we've built here is by hooking together two glucose monomers, we have created what's known as a disaccharide. It's a disaccharide because it contains two sugar monomers. And that particular disaccharide, does anyone know what it is? Do you know what it is if you put together two molecules of glucose? It's a disaccharide named maltose. Maltose. Right, so maltose is commonly found in the seeds of plants and things like that, and maltose is made by simply hooking together two glucose molecules by dehydration synthesis. So that is a disaccharide, and when we're hooking together, when we're splitting out water to create a covalent bond that joins together two monosaccharides, we refer to that type of bond as a glycosidic bond. That is called a glycosidic bond. So when you hook together monosaccharides to make polysaccharides, we call the bonds that you form through dehydration synthesis, we call those bonds glycosidic bonds. Okay, you can do the reverse, right? So here we have our glycosidic bond holding together the two molecules of glucose. If we add water and we put the water back in here, you will split them apart into two individual glucose molecules again, and that is called hydrolysis. Hydrolysis. Okay, so by the way, dehydration synthesis and hydrolysis will both happen spontaneously without any help, but they will happen much, much, much faster if we have enzymes that are there specifically to mediate those reactions. And in the human body, we have both types of enzymes. We have in certain places of the body, well, Humans don't actually have any enzymes that put together glucose into maltose. Only plants have those enzymes. But humans have lots of different enzymes that are made to put together monomers, and they have lots of different enzymes that are meant to take monomers apart through hydrolysis. Okay, so let's look at the carbohydrates. Let's now look at the 4 different classes of macromolecules, starting with the carbohydrates. Carbohydrates are commonly referred to as sugars or saccharides. That's a little bit of a misnomer because the word saccharide literally means sweet to the taste. And not all carbohydrates are sweet. For example, starch is the main constituent or the main component of rice and potatoes. And you know that potatoes and rice are not sweet. They taste, well, they don't taste, I don't know what you describe their taste as, but they certainly don't taste sweet. So all of these things, but nevertheless, starch is classified as a saccharide, but it's not sweet. So in fact, most of the carbohydrates are not sweet at all, but nevertheless we call them sugars or saccharides. All right, so polysaccharides are built, if you take, we start out with monosaccharides. These are the basic simple units that you put together to make larger sugars, larger carbohydrates. If you put together 2 monosaccharides, you've created a disaccharide. What do you think you would call it if you put together 3 monosaccharides? You're right. If you guessed that you would call that a trisaccharide. If you put together 4 monosaccharides, you would call that a tetrasaccharide, although that word is rarely ever used. And if you put together an indeterminate number of monosaccharides, if you put together just a chain of monosaccharides, we generally just call that a polysaccharide. A polysaccharide. So poly meaning several. Okay, now, are they hydrophilic or are they hydrophobic? Do they dissolve in water easily? Well, in fact, for carbohydrates, it depends on the size. So small carbohydrates tend to be soluble, so they're hydrophilic if they're small. The bigger they get, the less hydrophilic they tend to be, and so large polysaccharides tend to be hydrophobic. They do not dissolve. As I mentioned, starch is a polysaccharide that's made of, it's a polysaccharide that's made of glucose. It's a large polysaccharide made of lots and lots and lots of glucose molecules hooked together one by one, one after the other in tandem, as it's called. They're hooked together one after the other in tandem by glycosidic bonds. And you're familiar with starch because it's the main constituents of rice and potatoes, for instance. And you know that if you put potatoes into a pot of boiling water, they do not dissolve. They don't dissolve. And that's because starch is not hydrophilic. Starch is a hydrophobic molecule. But you do know, for instance, that if you put sucrose into water, it will dissolve. Sucrose is the carbohydrate that we call just ordinary table sugar. So when you have a cup of tea or a cup of coffee and you put sugar in it, what you're putting in is not just any sugar; it's a specific sugar called sucrose. And sucrose is a disaccharide that's made of 2 monosaccharides. One is called glucose and one is called fructose. And that dissolves, sucrose dissolves very easily in water because it's a disaccharide, so it's made of only 2 sugar monomers. It's made of only 2 monosaccharides, and so it tends to be hydrophilic. Okay, the sugars, the carbohydrates, not all of them are sweet. You can recognize whether or not we're talking about a carbohydrate because most of them end in the word ose, right? So glucose, maltose, cellulose. Starch is an exception because starch is not really the word that you're supposed to use. The starch is the common name for a polysaccharide called amylose, which also ends in ose. So usually you can recognize the fact that we're talking about a carbohydrate if it ends in ose. Right now, generally, we classify the carbohydrate monomers. We classify the monomers based on how many carbons they have. Okay, so glucose, for example, is a carbohydrate that has 6 carbons. And if you have any monosaccharide that has 6 carbons is given the general name a hexose. So a hexose is the generic name that you give to a monosaccharide that contains 6 carbon atoms. Okay, probably the most important hexose that we will ever study in biology is glucose, which is it is a hexose and it is the main source of energy in the human body. Glucose, right? If you have a monosaccharide that's composed of 5 carbon atoms, what do you think you would call it? If you guessed a pentose, you would be correct. So the word pento is a Greek word meaning 5, right? So in the United States, for instance, the big military building where they have the headquarters for the American military is called the Pentagon because it has 5 corners. Okay, so the most important pentose that we would ever study in biology is called ribose. Ribose is the backbone. It's the main constituent of the nucleic acids that are put together to form DNA and RNA. Okay, so generally a carbohydrate monomer that has 6 carbons is called a hexose. The most important one that we'll ever learn about in this course or any other biology course is glucose. And if a carbohydrate monomer has 5 carbons in it, if a carbohydrate monomer with 5 carbons, it's called a pentose. The most important one that we'll learn about is ribose. All right, so let's look at some of the common carbohydrates. Again, the simplest unit is referred to as a monosaccharide. One of the 2 most important monosaccharides that you should learn are glucose and ribose. If you put together 2 monosaccharides, you have a disaccharide. 2 of the important disaccharides that you should learn about are maltose and sucrose. There's another one called lactose that we haven't discussed yet. It's over here on the side list, lactose. Okay, so, and then finally, if you put together a lot of monosaccharides, you have a polysaccharide, which is a long chain of monomers. Okay, so the 2 most important polysaccharides that we learn about are amylose, which is commonly called starch, and cellulose, which is the main structural material that plants are made out of. So for instance, when you eat a plant, the edible parts of the plant, the parts that you can digest, are made of amylose, starch. There's another component called amylopectin as well. We won't go into great detail about that, but amylose and amylopectin are the 2 main edible parts of a plant. So if you eat a plant and the parts of it that you can digest are generally made of amylose and amylopectin. So potatoes are made primarily out of amylose and amylopectin. Rice is made primarily out of amylose and amylopectin. Okay, now the parts of the plant that you can't eat, such as the stem or the stalk, are made of cellulose, which is another form of glucose, a slightly different form of glucose. It's a polymer made of beta glucose instead of alpha glucose, and we'll discuss what that means. All right, so ribose and glucose look like this. So as I said, as advertised, ribose contains 5 carbons. It also contains 5 hydroxyl groups, and then glucose has 6 carbons, and it has 6 hydroxyl groups. But notice that the hydroxyl groups are not all on the same side. All right, so this is a branch of chemistry called stereochemistry, and so we have to worry about, if we were really studying chemistry seriously for this course, you would have to learn about what it means when the hydroxyl group is on one side versus the other. But this is not a serious chemistry course, this is a biology course, so don't worry about it. Okay, so now this is what ribose and glucose look like when they're in their linear form. But generally what happens is this double bonded oxygen here will usually come around and react with one of these hydroxyl groups to form a ring structure. The same thing is true for glucose, where this double bonded oxygen will swing around and react usually with this hydroxyl group and form a ring structure. And so glucose and ribose are usually found as ring structures, not as linear molecules. So they usually look like this. Now, if you're going to be a biologist or even a biochemist, you have to memorize the ways that we specify which ones of the carbons that we're talking about in these ring structures. So the way you distinguish one carbon from another, we have labels for we have to have a name for every carbon that you find in this ring structure. And here's how you define the name. Okay, so I'll just take glucose here as the example. Here's the oxygen, right? So the oxygen that's holding this ring together. You look at that, and then you move clockwise around the molecule and give the carbons that you run into numbers, and you call them 1 prime, 2 prime, 3 prime, 4 prime. So this is the 1 prime carbon which you find in glucose. This is the 2 prime carbon. This is the 3 prime carbon. This is the 4 prime carbon. And this is the 5 prime carbon. And this is the 6 prime carbon. Right? So you start from the oxygen, and then you go clockwise around the ring, and you give every carbon a prime number: 1 prime, 2 prime, 3 prime, 4 prime. All right, now let's try this over here at the ribose. Now here's the oxygen, and we're going to go around clockwise, and I want you to tell me what prime carbon this one is. Okay? If you guessed that that is the 2 prime carbon, you would be right. And this is ribose. And the difference between ribose and deoxyribose is that in deoxyribose, the 2 prime carbon is missing the hydroxyl group. It just has an H there instead of an OH, right? It just has an H. Right. Okay, that leaves us with, what's the number of this carbon? Okay, if you guessed that that is the 3 prime carbon, you'd be right. And when we put together polymers of DNA or RNA, it's DNA if we have deoxyribose with the 2 prime carbon missing the O, the oxygen, or it's ribose if it's not missing it. So messenger RNA, for instance, is made out of, it uses ribose as the backbone, whereas DNA uses deoxyribose as the backbone. Right. Now, if you put together these monomers, what you're actually doing is you're attaching the, there are usually 3 phosphate groups attached to the 5 prime carbon, and we split out we split out 2 of those phosphate groups and leave one of them in place, and that allows us to attach the 5 prime carbon of one of these riboses to the 3 prime carbon of the one in front of it, and that's how we make a polymer out of RNA or DNA. And we'll talk about that when we discuss the nucleic acids, but it's critical that you know how to number the carbons in the ring in order to tell which one attaches to which. Okay, so even though this is this is just elementary chemistry. For biology, for purposes of biology, we do need to know what the numbers are for the carbons in a ring structure, especially if we're talking about ribose. Okay, so ribose and glucose are both monosaccharides, but they are very important monosaccharides, much more important than any of the other ones that we learned. Okay, so glucose is a hexose that's used mainly for energy. Ribose is a pentose that's used to build DNA and RNA. Galactose is another hexose, and fructose is a pentose. Right? So galactose and fructose are 2 other monosaccharides that are important for biology. Okay, so glucose is created through photosynthesis in plants, right? And then if you put together a whole bunch of glucoses, you have starch, and then you can put all that starch into a storage nodule, which we call a potato. All right, so disaccharides. If you put together 2 monosaccharides, you have a disaccharide. Okay, so maltose is a disaccharide that's made from 2 glucoses. Sucrose is a disaccharide that's made from 1 glucose and 1 fructose. Remember, fructose is a pentose and glucose is a hexose. Lactose, where do you find lactose? You find lactose if you guessed that you find lactose in milk, you would be correct. Lactose is a carbohydrate that you find in milk. Milk is created through a process called lactation. Lactation is where the mammary glands of mammals produce milk. So the defining characteristic of a mammal is that all mammals produce milk and feed milk to their young. They produce milk in their mammary glands. In humans, the mammary glands are just called breasts, but all female mammals have mammary glands that produce milk to feed to the young. That's the defining feature of mammals. So therefore humans are mammals. All the other mammals have the same feature. Lions and tigers and dolphins and mice and rats are all classified as mammals, and they all feed milk to their young. So are pigs and cows, for instance. They feed milk to their young. All right, so you put these things together through dehydration synthesis, and you call a covalent bond between monosaccharides, you refer to it as a glycosidic bond, Okay, so here we see a glucose and a fructose that can be hooked together to create a glycosidic bond through dehydration, and we end up with the disaccharide called sucrose. That, of course, is table sugar that you put into your coffee or your tea. Lactose is found in milk. Lactose is glucose and galactose. It's a disaccharide made of glucose and galactose. All right, now when we're speaking of glucose, glucose actually exists in two very slightly different forms. One is called alpha glucose and one is called beta glucose. Notice that the only difference between alpha glucose and beta glucose is whether or not this hydroxyl group is above or below the plane of this ring structure. So this ring structure is meant to be flat. This is this particular type of drawing is called a Fisher projection, and you see where this line in front here is dark and thick. That thick dark line is meant to indicate that part of the ring is closer to you than this part back here. And so we're supposed to pretend that this ring is flat, and the flat part is, you're looking at it edgewise, and this dark line represents the edge of the plane that's closest to you, and the thin line back here represents the edge of the plane that's farthest away from you. Okay, so these are meant to be ring structures, and when you see here, for instance, this hydroxyl group is meant to be below the plane, below the ring, and this hydrogen is meant to be above the ring. Right? So the only difference between alpha glucose and beta glucose is whether this hydroxyl group here is below or above the ring, and that's the difference between alpha and beta glucose. It's a very minor, it's a very slight, subtle difference in the structure, but that slight difference in structure has very profound consequences, because humans and all the other animals on the planet have digestive enzymes that are able to hydrolyze polymers that are made from alpha glucose, but none of us can break down or hydrolyze polymers that are made from beta glucose. That's why we can't digest wood, because the primary constituent of wood is cellulose, and cellulose is a polymer that's made from beta glucose. Starch, potatoes and rice, amylose and amylopectin is made from alpha glucose. It's a polymer made of alpha glucose, and all the animals on Earth can digest alpha glucose. All right, there are only two types of creatures on Earth that can digest cellulose. One type is fungi. Right, so molds or mushrooms, for instance, are an example of fungus. So sometimes you see mushrooms growing on dead trees, and that's because they have the enzymes that are needed to break down or hydrolyze cellulose. The other organisms that can eat cellulose are certain bacteria. So there are certain fungi, certain molds, and certain bacteria that produce enzymes that break down cellulose. Those enzymes are called cellulase enzymes. And, well, you might say, well, what about cows? And what about other ruminant animals that can eat leaves and grass? And, cows and goats and deer, all of those animals can eat leaves and grass are made primarily out of cellulose, and you can feed, anyway, so these animals can eat it. How is that possible when you just told me that only fungi and bacteria can eat cellulose? And the reason is because those animals have two stomachs, and one of the stomachs is filled with symbiotic fungi and bacteria. And there are some protozoa, some protists, that can digest cellulose as well. Sorry, so there are three organisms on Earth. There's some fungi, some bacteria, and some protists that are able to digest cellulose because they possess cellulase enzymes. Okay, so it's not actually the cow that's able to digest the cellulose, it's the bacteria inside the cow's second stomach, which is called a rumen stomach. So the rumen stomach, a cow has a regular stomach, and then it has a rumen stomach, which is filled with symbiotic bacteria that are actually breaking down and digesting the cellulose for the cow. Okay, so amylose, also known as starch, is a polymer of alpha glucose. Okay, cellulose is a polymer of beta glucose, which is created by plants. And humans create a long polymer of alpha glucose, which is called glycogen. So now this is important because we're talking about humans now, and this is a human anatomy course. So glycogen is a polymer of alpha glucose, which is highly branched. It's a highly branched polymer. And it is the storage form that humans store their spare alpha glucose in. So when we eat food, when we eat sugars that can be broken down into glucose, some of the glucose goes into our blood, but the remainder of it is converted into glycogen and stored in the liver and also in the muscles. Right? So all humans have a supply of spare glucose that's stored in the liver and the muscles. The glycogen that's stored in the liver contains about enough glucose to live for 1 day. So you might have heard of these starvation diets where you're supposed to lose weight by starving yourself. Well, generally those types of diets don't work because your body, when your body doesn't have food, it goes after the spare glucose, the spare glycogen first. And so you would have to starve yourself for a whole day before the body even ran out of the glucose that it's storing in the liver. And then and only then would it start to go after the adipose tissue. Adipose tissue is where fat is stored. Right? So if you're using a starvation diet, you'd have to starve yourself for more than a day before your body even started to break down any of the fat. So most of us have enough glycogen in our liver to live for a whole day easily. Maybe even a day and a half or two, and then only then would our body start breaking down the fat, which is stored in a type of tissue called adipose tissue. All right, so here is human glycogen, which is being stored in the liver as well as the muscles. Amylose is a plant storage material that plants store in things like corn kernels on corn and rice grains on rice plants, as well as tubers in potatoes, right? So when we eat glucose, we're basically stealing the plant's spare glucose supply. And cellulose is used, the main constituent of wood and straw and the stalks and leaves of plants is mainly cellulose, which is made of beta glucose. Okay, so to summarize carbohydrates, we learned about mono, di, and polysaccharides. We learned about pentoses and hexoses and trioses. We learned about the fact that you can tell that you're talking about a carbohydrate when you hear that it has ose on the end. Amylose, glucose, cellulose, and so on. They usually form a ring structure. There's a difference between alpha and beta glucose. Cellulose is made of beta glucose, and we can't digest it because we don't have the enzymes that can hydrolyze it. But we have enzymes that can hydrolyze polymers made of alpha glucose. These sugars tend to be hydrophilic when they're small and tend to become more and more hydrophobic and insoluble the larger they get. Okay, so some of the very important carbohydrates are glucose and glycogen. Glycogen is the branched polymer of glucose that humans store for spare energy. Lactose is a sugar that humans produce in milk as well as all the other mammals. Amylopectin and amylose are two polysaccharides of alpha glucose that are used as storage material by plants, and when we eat them, they are broken down and put into our blood, and then some of it is polymerized into glycogen and stored in our liver. All right, let's move on to the second class of macromolecules, the lipids. Lipids are commonly known as fats, oils, and waxes. You know that fats, oils, and waxes do not dissolve in water, therefore lipids are hydrophobic. They are made of hydrocarbons. If you take a long chain hydrocarbon, in the last lecture we talked about saturated versus unsaturated hydrocarbons. So if you take a long chain that just consists of carbons and hydrogens, and it may be saturated, meaning it has only single bonds, or it may be unsaturated, meaning that it has one or more double bonds. Either way, it's a long hydrocarbon. If you stick a carboxyl group onto the end of it, then we stop calling it a hydrocarbon, and or an aliphatic hydrocarbon. We don't call it that anymore. We call it a fatty acid. So we change the name. If you take a long aliphatic hydrocarbon, saturated or unsaturated, it doesn't matter. If you stick a carboxyl group onto the end of it, we call it a fatty acid. Okay, then fatty acids are the monomers that are used to make up lipids. They tend to be almost exclusively hydrophobic. There's one exception, which is called phospholipids, and we'll get into that. Phospholipids are the main constituent of the cell plasma membrane, which we'll talk about later. Okay, so the monomers are long hydrocarbon chains. If you put a carboxyl group onto the end of a hydrocarbon, you rename it as a fatty acid. Those fatty acids are sometimes saturated or sometimes unsaturated, depending on whether they have just single bonds or whether they have one or more double bonds. And they're commonly called fats, oils, and waxes. All right, now one of the main forms of lipids that we have in the human body are lipids called the triglycerides. All right, so glycerol is an alcohol that has 3 carbons and 3 hydroxyl groups. If you attach, by the way, carboxyl groups will react with hydroxyl groups, if you remember your chemistry. Hydroxyl groups will react with carboxyl groups to form a link called an ester link. So if you react glycerol with 3 fatty acids, it will form what's known as a triglyceride. And this is what you find inside adipose tissue. Adipose tissue is made up of cells that are commonly called adipose cells. They're technically called adipose cells or adipocytes, but they're commonly called fat cells. Right? So fat is actually adipose cells that are filled with triglycerides, a particular fat called triglyceride. Okay, here we have glycerol. Notice that glycerol has 3 carbons and 3 hydroxyl groups. Right, so now we're going to see what happens if we react these 3 hydroxyl groups with 3 fatty acids. Okay, here's a fatty acid. Notice that on the end of the fatty acid, so we have a long hydrocarbon chain here, and on the end of the fatty acid we have a carboxyl group. That's a characteristic of an acid, isn't it? And over here we have a hydroxyl group, which is a characteristic of an alcohol. So glycerol is a common alcohol that you find in the body in the form of fat. And what we do is we take the OH from the carboxyl group of the fatty acid, and we react it with the H from the glycerol, and we make an ester link. Right, so we join those together by splitting out water, the same way that we did with polymerizing glycosidic bonds. Okay, if we do this all 3 times and attach 3 fatty acids to the glycerol, we will have created a triglyceride. So here we have a diagram of a triglyceride. So there is the original glycerol, and here are the 3 fatty acids, and here are the ester links. This is called an ester linkage. Right? And that is triglyceride. And so fat tissue on the body is made of cells that are filled with this stuff. Fat cells are filled with triglyceride. Okay, now triglyceride is, fat tissue. When you make butter, it's basically the triglycerides that has been boiled down and recovered from animal fat. Right, so you can't be a vegetarian and eat butter, because butter comes from the triglycerides of animal fats. What's the purpose of triglyceride and adipose tissue? The main purpose for humans and other mammals is that it allows mammals to survive in a colder climate. Now, we don't do any dissection in Anatomy 130, but if you take Biology 120 or Biology 100, you get to dissect a rat. You get to dissect rats and frogs. And if you've done that, if you've taken either of those courses, or if you have a friend who's taken those courses, they can explain to you that when you cut open the frog, you find adipose tissue, but it's located inside. It's not located on the just under the surface. You know, you see little strings of adipose tissue inside the frog that are kind of hanging there like bunches of bananas. And the purpose of adipose tissue is to have a reserve supply of energy, right? But the energy, this reserve supply of energy, this adipose tissue is not just under the skin of the frog. It's located deep inside, and therefore it doesn't provide any insulation. Whereas when you cut open the rat, you see that the adipose tissue is located just under the skin. Therefore, it's wrapped around all the vital organs, and it keeps the vital organs warm because it's a good insulator, right? So mammals happen to have the adipose tissue close to the surface, and they can use it for insulation. And that allows mammals to live in a much colder climate than frogs, which are amphibians, for instance. So frogs and reptiles can't live in cold climates because they are not insulated by their adipose tissue. But mammals are. So that has allowed the fact that mammals, including humans, have adipose tissue close to the surface, just under the skin, allows them to live in colder climates. Okay, what about oils? The difference between fats and oils is that fats are made with saturated hydrocarbons, and oils are made with triglycerides that are made with fats are made of triglycerides that have saturated fatty acids, and oils are made of triglycerides that have unsaturated fatty acids. Now, what that means is that if you have unsaturated fatty acids, they're not symmetrical, they're bent. You remember how we showed that mostly it's a cis double bond? So having a cis double bond puts a bend into the hydrocarbon chain, if you remember that from the last lecture, and that prevents the fatty acids from being able to stack on top of each other. It prevents them from being able to stack one on top of the other. And if that happens, if you can't stack one on top of the other easily, you do not form a solid at room temperature. But if the triglycerides are made entirely of saturated fatty acids, they can stack one on top of each other quite easily. They're symmetrical, and symmetrical things that are stacked one on top of each other tend to form a solid at room temperature. So you know that butter is a solid at room temperature. It's certainly a solid if you have butter in the refrigerator. But if you heat it up a little bit in the frying pan, it does melt. Oils are liquids at room temperature because they have a lower freezing point, right? So they are liquids at room temperature. But if you put oils in the refrigerator, they will become a solid. Many of them will become a solid. So there are many oils that if you put them in the refrigerator, they kind of become a solid. Okay, what about waxes? Waxes are things that are made by the body. There's certain glands that produce waxes. So you know, for instance, that the human body makes wax and puts it into the ear, the inner ear canal. And you know that bees have similar glands that make waxes that are located in their mouth. So they produce these waxes in their mouth, and then they weave that into a honeycomb. Right, so a wax is a long, complicated fatty acid that's attached to a very long, complicated alcohol. Right, so it's not glycerol attached to 3 fatty acids. It's one very long fatty acid attached to one very big, very long alcohol. And that molecule, this wax, tends to be a strong solid at room temperature, but you know that you can melt beeswax at a higher temperature. But at room temperature or below, wax is a very strong solid that is very hydrophobic. Okay, so here we have oils that are made of basically triglycerides that have unsaturated fatty acids, versus waxes that are made from one large fatty acid and one large alcohol. Okay, so the term saturated, of course, means that there are no double bonds in the carbohydrate, and the term unsaturated means that there are some double bonds in the fatty acids. And if you have double bonds, they won't stack symmetrically, and then they tend to be liquids at room temperature. We learned in the last lecture that these double bonds can be either the cis or the trans configuration. Can you remember how you create the trans configuration? Do you remember which one is bad? It actually says there, so it's not much of a challenging question. The trans configuration is bad for you because it doesn't occur in nature. But the cis configuration puts a bend into the fatty acid chain, and that's what stops them from stacking symmetrically. Okay, so here we have butter that's made of saturated fatty acids, saturated triglycerides, and it's a solid at room temperature because these fatty acid chains can stack on top of each other easily, making it easy to form a solid rather than a liquid. Liquids have at least one of the chains unsaturated. So here's a cis double bond that creates a bend. If this is bent, it can't stack on top of other triglycerides very easily, and it forms a liquid at room temperature. Okay, you may know that margarine is made made of what's known as partially hydrogenated vegetable oil. Right, so what they do is, okay, so the vegetable oil is an oil because it has double bonds. What if we broke all those double bonds, or what if we broke as many as we can and turn them all into single bonds? The vegetable oil will go from being a liquid into a solid. That's true. So what they do is they bubble hydrogen gas through the oil, which has the effect of breaking the double bonds, most of them, and then converting the vegetable oil from a liquid to a solid that you can spread onto your toast, for instance. Right now, the problem is that when you hydrate or hydrogenate a vegetable oil, you don't succeed in breaking all of the double bonds, and in fact, the way you break a few of the bonds, it actually ends up creating a few trans double bonds. Right, so the vegetable oil started out with a lot of cis double bonds. You hydrogenated it, you broke most of them, but you converted some of them into trans double bonds instead of cis double bonds, and trans double bonds are very bad for you. Okay, so here's an unsaturated fatty acid with a cis double bond. Right, here's a unsaturated fatty acid with a cis double bond that puts a bend into the hydrocarbon chain. Here's a trans double bond, which is bad for you. So for reasons we don't quite understand, the human body cannot handle trans unsaturated fats, and so they end up clogging up the arteries and causing arteriosclerosis and heart disease. In fact, this is my strong opinion and the opinion of most people who know about this. Trans unsaturated fats and hydrogenated vegetable oils should be illegal because they are causing heart disease. They're causing people to have heart attacks. However, the people that make margarine, so margarine is very bad for you, so you shouldn't eat margarine. If you're a vegetarian and you don't want to put butter on your toast, that's fine. What you can do is, don't laugh, this is perfectly serious. What you can do is you can pour olive oil onto your bread, or you can pour a few drops of olive oil onto your toast and smear it around with a knife, and then you can eat it. In fact, the ancient Romans didn't put butter on their bread, they put olive oil on their bread, and they never complained about it. They thought it was quite nice. In fact, so I do that, and a lot of people do that. You can just put olive oil onto your toast, a little bit so that it doesn't get soggy, but it takes the place of butter, right? And you never have to worry about, margarine. Sometimes if you've had it in the refrigerator, it's hard to spread it because it's a solid. But the olive oil, you can keep it at room temperature, and then you can just pour a few drops onto your bread. Okay, so hydrogenated vegetable oils and margarines should be illegal because they are causing heart disease. But the reason it's not illegal is because the people who make margarine basically pay bribes to the politicians so that they won't make it illegal. So the medical people tell the politicians this is an actual health problem. Meanwhile, the business industry says, yes, but if you force us to stop making it, we'll go broke. And then the politicians say, well, I don't really know what to do. Keep on making it for now, and we'll think of something. And so bribes, when I say bribes, I'm being facetious. I mean donations to political parties and say, okay, we're going to give you, that in Canada and Western Europe and North America, that's referred to as lobbying. Lobbying is essentially a sanitized version of bribery, where individuals or companies say to politicians, "We'll donate money to your political campaign if you do what we ask." And so, if you want to be reelected, then you should do what we ask, and then we'll give you the money. If you don't do what we ask you to do, we'll give the money to your opponents. And so they usually do what they want. So one of the things they've done is, margarine should be illegal. Hydrogenated vegetable oil should be illegal. Much of the fast food, French fries that you buy at McDonald's and Wendy's, and chicken McNuggets that you buy at McDonald's and Wendy's and things like that, those things are deep fried in partially hydrogenated vegetable oil, and so they have transunsaturated fats which cause heart disease, and that should be illegal. The reason why they deep fry them in partially hydrogenated vegetable oil is because it doesn't spoil as fast. Right, so the amount of money that the fast food industry is saving by using, if they deep fried the French fries and the chicken McNuggets in olive oil, just regular, or canola oil, just regular oil, they would have to change the oil in the deep fat fryer more often. So they would lose a little bit of money, but society would gain because fewer people would be having heart attacks. So fewer people would have heart attacks. The fast food industry might have to accept a slightly smaller profit margin, but deep frying things in partially hydrogenated vegetable oil should be illegal, and then margarine should be illegal. Another thing that should be illegal is cigarettes, because it's proven beyond a shadow of a doubt that cigarettes cause lung cancer, as well as emphysema and chronic obsessive pulmonary disease, which we'll talk about later when we get to the respiratory system. That should be illegal too. But again, the businesses bribe the politicians by lobbying them, donate money to political campaigns, and then they give them a reason to believe that they're doing the right thing by saying, well, we're employing a lot of people. So if you made this illegal, if you made cigarettes and margarine illegal, a lot more people would be unemployed. So sure, you don't like the health risk that these products are posing, but we know that you want to keep people employed. So why don't you just tell yourself that? Right? So a lot of that's called cognitive dissonance. Cognitive dissonance is where you do something selfish, but you find some noble reason to believe that you're actually doing something altruistically. You're doing the right thing for a noble reason, because you don't want to believe that you're actually not a good person who's doing something for an ignoble reason. All right, that was my diatribe. I'm sorry about that. Okay, so now we'll move on to something called the omega nomenclature for fatty acids. You may have heard the term omega-3 fatty acids or omega-6 fatty acids. So there is a way of telling somebody which carbons have double bonds in a fatty acid, and this is the omega system. Okay, so the Greek letter omega, ancient Greek letter omega, is the last letter in the Greek alphabet. So if you know your chemistry, usually when you're counting carbons in an aliphatic hydrocarbon chain, you count the first carbon as number 1 and then number 2, 3, 4, 5. In the omega system, you forget about the number of the first carbon. You label the last carbon in the chain as the omega carbon. Right? So by the way, what's the first Greek letter? What's the first letter in the Greek alphabet? Right? The first Greek letter is alpha. Second Greek letter is beta. So alpha is the first letter in the Greek alphabet. Omega is the last letter. So what they do in this system of naming unsaturated hydrocarbons, you name the last carbon the omega carbon. And then what you do is you count backwards from the omega carbon inwards. You count backwards until you hit a double bond. If you count 1, 2, 3 carbons, and then there's a double bond at the third carbon, you call that an omega-3 fatty acid. If you have to count 6 before you hit a double bond, so you count omega 1, 2, 3, 4, 5, 6, and then there's a double bond, you call that an omega-6 fatty acid. Okay, so here we have a fatty acid which is referred, the long name is alpha linoleic acid, and it is an example of an omega-3 fatty acid. So what we do is we forget about carbon number 1. The organic chemists would normally label that. Where the carboxyl group is, they would label that as carbon number 1. We're not going to do that. We're going to forget about that and just label the last carbon the omega carbon. And then, so that's the omega carbon is number 1, then you count backwards to carbon number 2, and then carbon number 3, and at carbon number 3 we have a double bond. So we refer to alpha linoleic acid as an omega-3 fatty acid. It's an omega-3 fatty acid. If that double bond had been further down until we had to go 6 to find a double bond, then it would be an omega-6 fatty acid. All right, now, I said that lipids are mostly hydrophobic. There's one exception, and that is the phospholipids. Okay, in a phospholipid, you take triglyceride, and remember that a triglyceride has 3 fatty acid chains that are attached to the 3 carbons in glycerol. You take one of the fatty acids off and replace it with a phosphate group, and a phosphate group is highly hydrophilic. A phosphate group likes to be dissolved in water. So what you've created is a molecule that has 2 fatty acid chains on one end that are very hydrophobic, and a phosphate group at the other end which is very hydrophilic. So you've created a molecule that's hydrophobic at one end and hydrophilic at the other end, and we call a molecule we say that a molecule like that is amphiphilic. So an amphiphilic molecule is a molecule that's hydrophobic at one end and hydrophilic at the other. Okay, so phospholipids are amphiphilic, and they create something called a phospholipid bilayer. They are the main constituent of the plasma membrane, which is the outer part of a cell. It's the outer boundary of a cell. So the outer boundary of a cell is referred to as the plasma membrane, and it's made of phospholipids. Okay, so here we have a triglyceride on the left. We remove one of the fatty acids, and then we replace it with a phosphate group, which is hydrophilic. Right, there's your phosphate group, and then we take one of the triglycerides, and instead of having we take one of the fatty acids, and instead of having it be saturated, we have it be unsaturated. Right. So the difference between triglyceride and a phospholipid is that a triglyceride has 3 fatty acids attached to the glycerol, and generally they are all saturated. Whereas phospholipids have 1 phosphate group and 2 fatty acids, and of the 2 fatty acids, one of them has at least 1 double bond. Okay, so what happens if you put a bunch of phospholipids in water, the fatty acid tails don't want to be in the water, and the phosphate groups do want to be in the water. So how do you resolve that? Well, what it does is it makes 2 layers where the fatty acids are facing inwards towards each other. Right? So here you see that these little spheres at the top here are meant to be phosphate groups, and these little yellow tails are meant to be the fatty acid tails. And you see that there are 2 layers. There are 2 layers of them, and they are arranged in a way that the phosphate groups are facing the water outside, and the fatty acid tails are facing each other on the inside. And that's called a phospholipid bilayer, and that is what forms the outer boundary of all cells. Right? So all cells have a boundary on the outside, their outer shell, if you want to call it that. Their outer shell is made of a phospholipid bilayer. All right, now steroids. There is a chemical compound called a steroid, which is it's a hydrocarbon structure. Basically, all you do is you take a fatty acid, and instead of attaching it to glycerol, instead of leaving it as a chain, instead of leaving it as an aliphatic chain of hydrocarbon, you bend it into a ring structure. Okay, so you take one of the fatty acid chains and you bend it into a ring structure, and you make something called cholesterol. Cholesterol, because it's a ring structure, is not flexible the way a fatty acid is. Now, if you insert cholesterol into the phospholipid bilayer that I just mentioned, you will make it less flexible and more rigid and less fluid-like. Right? So cholesterol is used to reinforce and strengthen the plasma membrane of every cell. So we've heard that, we've heard in popular media, we've heard that cholesterol is supposed to be bad for you. In fact, cholesterol is not bad for you. It's essential. Humans need, and other animals need, lots and lots of cholesterol because they use it in every single cell. The cells would be too weak if not for the fact that they were reinforced by cholesterol. So cholesterol is not bad, but simply having too much cholesterol floating around in the blood is bad. Having too much cholesterol in the blood is bad. Okay, so cholesterol is where you take a fatty acid, bend it into a ring structure, and then insert it into the phospholipid bilayer to give it more strength. Okay, so here we take, if you look on the top right, you see that there's a fatty acid chain. If you take that off and bend it into a ring structure like this, you've created cholesterol. Right. Now, if you take cholesterol, basic simple cholesterol, which is found in every cell, which is found in large quantities in every cell, if you take cholesterol and you decorate it a little bit, you change it a little bit here and there, you will create things that are called steroid hormones. Steroid hormones are produced by the endocrine system, and they are used to affect changes in the body. They cause changes in the body. So the steroid hormones are used as signals that are sent from one organ to another to tell it to do something, right? And so this, as I just showed you, on the right we have cholesterol, right? And cholesterol has 4 rings, 3 6-carbon rings and 1 5-carbon ring. If you modify it slightly, you get this molecule that you see on the left, which is called testosterone. And testosterone is a hormone that causes boys to develop into men. It causes the formation of the male body shape. That male body, for instance, has more, is taller and bigger and has more muscle tone than the female body because the male body produces more testosterone. Okay, so testosterone is a steroid hormone. Progesterone is a steroid hormone that women produce. There are other steroid hormones that either men or women produce as well. And the steroid hormones are a group of signals that are sent around the body that are originally created by modifying cholesterol. So you simply modify cholesterol, change a little bit here and there, and you can create any one of the steroid hormones that are used as signals. Okay, to summarize lipids, they're usually hydrophobic except for phospholipids, which are amphiphilic. They are built from fatty acids and alcohols, particularly glycerol. So if you put 3 fatty acids together with glycerol, you've created a triglyceride. Triglyceride is what we commonly call fat. That fat is stored in adipose tissue, and it's used as an insulation. It's also used as an energy, as a supply of spare energy. Humans generally carry spare energy in 2 forms. Number 1, I talked about when we talked about carbohydrates. What is the spare form that we store glucose in? Do you remember? We store spare glucose in the form of glycogen, and it's stored in the liver and the muscles. Right? So we generally have enough glycogen in our body, particularly in the liver, to live for about 1 day without eating. You need to drink, right? You could easily survive for 1 day without eating, and you could survive on the glycogen being converted into glucose and put into the blood. But you need to keep on drinking because in order to break glycogen down into glucose, you have to hydrolyze it, don't you? So you need water to hydrolyze things. So people can live for a day or 2 just on glycogen without food. They can live on just glycogen, but you do have to drink. If you don't drink water, you'll die very fast because you need water for most of the metabolic functions in your body. Okay, so human body has 2 forms of energy storage. One is the glycogen, which is stored in the liver and in the muscles, and the other is the adipose tissue, where the triglycerides. If you break down glycogen into glycerol, it generates a certain amount of energy. But if you break down triglycerides, they can be converted into glucose, but it actually produces quite a bit of glucose. So there is actually a lot more spare energy stored in adipose tissue than there is in glycogen. There's a lot more spare energy stored in triglycerides than in glycogen. Right? So the average human, if you were starving but you didn't have anything to eat but you had water to drink, the average human could live for 1 day on the glucose and the glycogen that's in their liver, and then the average human could probably live for a couple of weeks, perhaps even a month, on the stored adipose tissue. Obviously, that depends on how much adipose tissue you have. If you're middle-aged like me and you have, probably 20 kilograms of spare adipose tissue, you can live a lot longer than if you don't have any. Like supermodels that starve themselves and exercise all day have very little triglyceride, so they wouldn't live very long if the food suddenly ran out. Okay, so the triglycerides and glycogen are the two forms of energy storage for the body that are relatively easily converted back from a storage product into an energy product. It's easy to convert glycogen back and forth between glycogen and glucose. The conversion is very easy, and so that's what your body does first, right? So if you run out of food, the first thing your body does is start breaking down the glycogen. You can live for a day that way. Then if you still haven't got any food, your body will break down the triglycerides, and then you can live for 2 weeks or even a month that way. And then when the adipose tissue runs out, unfortunately, your body starts breaking down proteins. You can break down proteins and use the proteins and use them to generate energy. But the problem is that we don't have any spare proteins in our body. There's no such thing as a spare protein. So once your body if in the state of starvation, once your body starts to break down proteins, that's generally terminal. That means you're about to die. You can live for a day or two on breaking down proteins, but not very long, because you don't have any spare proteins. So every protein that's taken apart represents a bodily function that you're losing. Okay, so lipids. We have triglycerides. We talked about phospholipids and how they're used to make the phospholipid bilayer. We talked about steroids and cholesterol, steroid hormones and cholesterol. And we talked about how to name omega-3 and omega-6 fatty acids. I should also mention the fact that theoretically, humans, at one time, we had enzymes that could make all of the possible fatty acids that we need to build all of our body tissues. However, over thousands and millions of years of evolution, humans have actually lost the ability to make several of the fatty acids that we need to live. And so we must get those fatty acids in our diets. And so that's what they mean when they try to sell you fish oil pills that are enriched in omega-6 and omega-3 fatty acids. They're talking about what are known as essential fatty acids. So the fact that we can't make all of the types of fatty acids that we need to live is why we need to eat some of them in our diet. We need to gain some of those fatty acids in our diet. And the fatty acids that we must get in our food are referred to as essential fatty acids. And many of them are omega-3 and omega-6 fatty acids. Okay, let's move on to what I think is a more interesting macromolecule, which is the proteins. Okay, so proteins, many of the tissues in the human body are made of protein. The muscle tissue in the body is made entirely of protein, mostly a protein called myosin. The fingernails and the hair are made of a protein called keratin. There's a protein called hemoglobin that carries the oxygen around the blood. Many of the other tissues in the body are made up of proteins that are called elastin, and a protein called collagen, and a protein collagen, elastin, and hydroxyapatite, and other, collagen, elastin, and fibronectin are three very common proteins that hold all the different parts of the bodies together. So, and then of course there are many, many proteins that are enzymes that catalyze the reactions that build all the other stuff that we're made of. Okay, so proteins are made of monomers that are called amino acids. I mentioned in the lecture, in the previous lecture, that amino acids are molecules that have an amino group on one side and a carboxyl group on the other side. If you put them together, through dehydration synthesis. You do this on an organelle, a little tiny organ in the cell called a ribosome. We'll be discussing ribosomes in detail later. Some of them are hydrophobic and some of them are hydrophilic. Generally, I just mentioned the fact that hair and fingernails are made of a protein called keratin, right? So hair and fingernails, hair is not hydrophobic. I mean, hair is hydrophobic. Obviously, when you go swimming, your hair gets wet and it likes to be wet. However, your hair doesn't dissolve in the water, right? So hair doesn't dissolve when you go in the water. That proves that the hair is actually hydrophobic. So keratin is what's known as a structural protein, and it tends to be it happens to be hydrophobic. There are other proteins like hemoglobin that are hydrophilic, that are meant to dissolve in the serum of the blood, in the blood serum. Right? So generally hydrophilic proteins are enzymes tend to be hydrophilic proteins because they have to be dissolved in the aqueous solution that is the cytoplasm, the inside of the cell. They need to be dissolved in the liquid inside the cell and catalyze the chemical reactions that are going on. And we generally refer to proteins that are hydrophilic and they're kind of blob shaped, they're kind of roughly round in shape. We call those globular proteins. And globular proteins they dissolve easily in water, and so most enzymes are globular proteins. All right, so enzymes catalyze all the reactions to make a lot of things in the body, and proteins are the only macromolecule that is actually encoded by the genes. So the DNA in the nucleus contains a bunch of genes, and those genes code for proteins. There are no genes that code for carbohydrates or lipids or anything like that. All right, so proteins are made from, you put together long chains of amino acids, and there are 20 different amino acids. The 20 different amino acids have different chemical properties, and so depending on which amino acids you put together and in what order do you put them together, that will determine the properties and the characteristics and the behavior of the protein that you build out of them. Right. Okay, so there are 20 amino acids. There are 20 different amino acids that we use to build all of the proteins that make up the human body. But actually, 9 of them we have lost the ability to make for ourselves. Right? So there are some organisms on Earth that can make all 20 of the amino acids. Humans, millions of years ago, used to be able to make all 20 of them. But over the course of evolution, we've lost the ability to make about 9 of them. So there are 9 amino acids that we must eat in our food or we die, and those are referred to as the essential amino acids. So I mentioned in the last section that there are essential fatty acids. We used to have the enzymes that can build those fatty acids. We've lost the ability because those genes have mutated over millions of years. And so we need to get those fatty acids in our diet. We call those essential fatty acids. And there are 20 amino acids. They are built, all of the amino acids are built by enzymes, and the genes that encode the enzymes that build 9 of those amino acids have been mutated in humans so that we can't make them anymore, and so we must get them in our diet. We must eat them. Okay, so as I said, the 20 different amino acids all have different chemical and biochemical properties. And depending on which amino acids you put together in which order, that will determine the properties of the protein. Okay, so enzymes are proteins that facilitate chemical reactions, including the chemical reactions that are necessary to build the amino acids. Okay, so amino acids, they usually end in "ene", right? So glycine is an amino acid, alanine is an amino acid, phenylalanine is an amino acid. They each have a name. All 20 of them have a name. So glycine and alanine are the full names for those 2 amino acids. But we also refer to them by a 3-letter code and by a 1-letter code. So can you guess what the 3-letter code is for glycine? It is GLY, right? What's the 3-letter code for alanine? Of course, it's ALA, right? So you can abbreviate glycine and alanine with GLY or ALA for gly or ALA. And each amino acid also has a 1-letter code, which is fairly easy to guess here. So the 1-letter code for glucose is G, and the 1-letter code for alanine is A. Right, now there are 20 amino acids, and there are some amino acids. There's another amino acid called glutamate, And so we use another letter. We have to use another single letter for glutamate because G is already taken by glycine. But if you take Biochemistry 201, you'll have to memorize the names of all 20 amino acids, which is not really as hard as it sounds. And also you'll have to learn the abbreviation codes, but that's all right. All right, so an amino acid has an amino group on one side and a carboxyl group on the other side, and then there are 20 different things that are called side chains, which is what gives each one of them the unique properties. They're all the same in the sense that they have an amino group on one side and a carboxyl group on the other, but they all have a different side chain. Okay, so the different side chains determine 20 different physical and biochemical properties. And then if you put these amino acids together one by one through dehydration synthesis on an organ, a little tiny microscopic organ called a ribosome, then you can create proteins. And when you put each amino acid together, you do so by splitting out a water molecule, and this time you create a covalent bond that is referred to as a peptide bond. You remember when we put together two or more monosaccharides, we call a covalent bond that's formed between two monosaccharides by dehydration synthesis, we call that a glycosidic bond. If we have two amino acids that are put together by dehydration synthesis, we call that a peptide bond. It's a covalent bond, but it's given the special name peptide bond. Okay, so here is a generic diagram showing you what an amino acid looks like. So on one end we have the amino group, that's the same in all 20 of them. And the other side we have a carboxyl group, that's the same in all 20 of them, and that's why it's called an amino acid. In the center we have something called an alpha carbon, and then we have a hydrogen. Okay, so far all of this stuff is the same in every amino acid. Finally, we have this thing which is called a side chain or an R group. It's called a side chain or an R group, and that is what's different in every amino acid. And that side chain or that R group is what determines the physical properties of that amino acid. Okay, so this diagram is from your OpenStax textbook, and it shows all 20 of the amino acids, and it shows what the side chain is. Okay, so here's glycine that we talked about before. Notice that this bit, the amino terminus, the alpha carbon, the carboxyl group, and the other hydrogen, the alpha hydrogen, are all the same. And the side chain in glycine is just an H. It's just another hydrogen. Right? So glycine is actually the smallest amino acid because it has the smallest side chain. Okay, next alanine. This part again looks the same, but over here we have a methyl group. Right, so the side chain for alanine is a methyl group. And here we have a slightly different side chain for an amino acid called valine. And then we have an even larger side chain for an amino acid called leucine, an even bigger one for an amino acid called methionine, and an even bigger one for an amino acid called isoleucine. Okay, now all of these side chains they become more and more hydrophobic depending on how big they are. So isoleucine is much more hydrophobic than glycine. Glycine is not hydrophobic at all, it's very hydrophilic, so it'll easily dissolve in water. All right, now these three amino acids, lysine, arginine, and histidine, they all have positively charged side chains, and we call them basic amino acids or positively charged amino acids. These two amino acids, aspartate and glutamate, have negatively charged side chains. In fact, it's a carboxyl group that's negatively charged. And so we call these acidic amino acids or negative amino acids. Okay, these, phenylalanine, tyrosine, and tryptophan, all have ring structures in their side chains. Ring structures, we learned in the previous lecture, they are called aromatic hydrocarbons, and so we call those three the aromatic amino acids. And then these other ones have various properties. One of the main properties is that serine, threonine, and where's the other one? I can't find it. Serine, threonine, and tyrosine is down here, sorry. They all have a hydroxyl group in their side chain. So there's a hydroxyl group there, there's a hydroxyl group there, there's a hydroxyl group there. So that means that those three amino acids have the properties of being an alcohol, which is the characteristic of a hydroxyl group. Okay, so we won't really go into all the properties, but of the 20 amino acids, but just bear in mind that if you, depending on which amino acids you make a protein out of and what order you put them together in, that is what will determine the properties of that protein. Okay, so if you put together two amino acids, you take the OH from a carboxyl group of one, and you take one of the H's from the amino group of another one, and you split out the water, you create what's called a peptide bond. Peptide bond is the special name that we give to a covalent bond that joins two amino acids together. All right, this is a protein. It's actually made of two chains of amino acids that are hooked together by something called a disulfide bridges. But notice that the particular sequence of this amino acid is on one chain we have glycine, isoleucine, valine, glutamic acid, glutamine, cysteine, alanine, serine, valine, cysteine, serine, leucine, tyrosine, glycine, glutamine, leucine, glutamic acid, asparagine, tyrosine, cysteine, and asparagine again. That particular chain of amino acids has certain properties, and then it's also attached to this chain that starts with phenylalanine, valine, asparagine, and so on. That's how you make a protein. And this particular protein, composed of those amino acids, is actually insulin. It's human insulin, which is a protein that diabetics have to inject themselves with. So this is the structure for insulin. It's a fairly small protein. When we have a small protein, we don't actually call it a protein. You call it a peptide. Peptide refers to a very small protein, but don't worry about that. Don't worry about that right now. Right, so this is a protein, insulin. And it's made up of these amino acids put together in that particular order. Okay, so what are the functions of different proteins? Some of them are enzymes that catalyze reactions. Some of them are structural, like keratin, for instance, that makes up hair and fingernails. Some of them are involved in movement, like myosin, the protein myosin that is the main constituent of muscles. Some of them are involved in transport, like hemoglobin, that is responsible. Hemoglobin is a globular protein that is responsible for moving oxygen around the body. All right, now on the subject of enzymes, right? Enzymes catalyze chemical reactions, and in biochemistry and in biology, we have a special name for naming the enzymes. What you do is you take the substrate. The substrate is whatever it is that the enzyme works on, right? So if you have an enzyme that works on cellulose, for instance, so the substrate that the enzyme works on is cellulose. Okay, if you have an enzyme that breaks down amylose, the substrate for that enzyme is amylose. All right. So the substrate is the thing that the enzyme works on. It either takes it apart or puts it together. And so the common way of naming enzymes is to take the name of the substrate and add A's to the end. Right. So cellulose, the enzymes that hydrolyze cellulose are called cellulase enzymes. The enzymes that break down amylose are called amylase enzymes. The enzymes that break down proteins are generally referred to as protease enzymes. Enzymes that break down lipids are referred to as lipases. Okay, now every enzyme has a set of optimum enzyme conditions. So every enzyme has a temperature that it works best at, a pH that it works best at, and a substrate concentration that it works best at. Okay, so in the human body, what temperature do you predict that the enzymes of the human body would work best at? Well, if you guessed 37 degrees Celsius, you'd be right, because that's normal human body temperature. So all of the enzymes in the human body have an optimum operating temperature of 37 degrees. If you lower the temperature below 37 degrees, they don't work as fast. If you raise the temperature above 37 degrees, they don't work as fast. Okay, what about most of the human body? Do you know what the optimum pH for enzymes around the body would be? It would probably be whatever the pH of the human body is. So most of the human body has a pH which we don't call it neutral pH. Neutral pH is what water is supposed to be in theory, which is 7. So 7 is neutral pH. the human body, most of the human body is slightly basic compared to water. So the human body is buffered to what's known as physiological pH. So there's a difference between neutral pH and physiological pH. Physiological pH is between 7.2 and 7.4. It's a range between 7.2 and 7.4. Okay, so there are most enzymes in the human body work best, they work fastest, and they work best at a pH of between 7.2 and 7.4. If you lower the pH below 7.2, they don't work as well. If you raise it above 7.4, they don't work as well. Okay, what about the stomach? Do you remember what the pH is of the stomach? Inside the human stomach, we have a pH of 2, and there are enzymes in the stomach that break down proteins, and that there's one enzyme, in fact, that breaks down proteins, and it is referred to as pepsin. So pepsin has an optimum pH of 2, so pepsin works best at pH 2. The human intestine is an alkaline environment, so it has a pH of about 8, and so there are enzymes in the human intestine that are breaking down the food that we've eaten, and they work best and they work fastest at a pH of 8. Okay, so let's practice naming these enzymes one more time. What do you call enzymes that break down cellulose? What do you call enzymes that break down amylose? What about proteins that break down proteins? Enzymes that break down proteins? Enzymes that break down lipids, and enzymes that break down or remove phosphate groups. Okay, let's have a look. You can stop the recording if you want to guess. So the answers are, we call an enzyme that breaks down cellulose, that hydrolyzes cellulose, we call it a cellulase enzyme. An enzyme that hydrolyzes amylose is called an amylase. Enzymes that break down proteins are generically called proteases, but some of them have specific names. So for instance, I just mentioned that there's a protease in the stomach called pepsin. So pepsin is a protease, but not all proteases are called pepsin. Pepsin is a specific protease. Okay, so proteins that break down lipids are generically called lipases. Now, if an enzyme's job is to remove phosphate groups from other proteins or from nucleic acids, it's referred to as a phosphatase enzyme. What about enzymes whose job it is to put phosphate groups onto proteins? If an enzyme whose job it is to put phosphate groups onto proteins are referred to as kinases. It's referred to as a kinase. So don't forget the word kinase. Okay, so to summarize, proteins are made out of 20 different amino acids. The 20 different amino acids have different chemical properties, and that's what determines the property of the protein that you make by putting several of them together. Okay, so these are the only macromolecules that are encoded by the genes, and some of those genes encode enzymes that mediate chemical reactions that can be used to create the other 3 classes of macromolecules. Okay. All right, let's finish off by looking at nucleic acids, RNA and DNA. Okay, so if you take ribose, we talked about ribose as a carbohydrate with 5 carbons, and we learned how to number those carbons. Okay, so if you take ribose and then you put onto carbon number 1, you put something called a nitrogenous base. A nitrogenous base. And there are 6 of them actually. 5 of them, sorry. There are 5 different nitrogenous bases that you can put on carbon number 1. The 5 different nitrogenous bases determine which nucleotide you will have built. So if you take a ribose molecule, you put one of 5 possible nitrogenous bases onto carbon number 1, and then you add 3 phosphate groups to carbon number 5, you will have built something called a nucleotide. And the nucleotides are the monomers that DNA and RNA are built of. So if you put together a long chain of nucleotides, you make either DNA or RNA, and that depends on whether you started out with ribose as the backbone or whether you had deoxyribose as the backbone. So DNA, deoxyribose, is what genes are made of. Ribose, just regular ribose, is used for messenger RNA to transcribe genes and convert genes into proteins later. Okay, so here we have ribose. Remember, carbon number 2 has the oxygen on it in ribose. Right? If you remove the oxygen from carbon number 2, then you have deoxyribose. Okay, and then if you take carbon number 1, this one, and you remove this hydroxyl group and substitute something called a nitrogenous base, and there are 5 of them to choose from. And then over here, if you take away this hydroxyl group and you add two phosphate groups, so you've taken this thing and you stick a nitrogenous base onto carbon number 1, and you stick 2 phosphate groups onto carbon number 5, you've created a nucleotide. Okay, so here's our original ribose or deoxyribose here, right? In this case, they've drawn in deoxyribose. And then on carbon number 1, we add a nitrogenous base. And then on carbon number 5, we add a phosphate group. In fact, what you do is you add 3 phosphate groups, and then 2 of them are removed when you fuse when you put together 2 nucleotides. Okay, so this is a nucleotide. That nitrogenous base, there are 5 of them to choose from. One of them is called cytosine, which is just commonly abbreviated C. One of them is called thymine, which is abbreviated T. One of them is called adenine, which is abbreviated A. One of them is called guanine, which is abbreviated G. And one of them is called uracil. Uracil is found in RNA, and uracil is the same basically as thymine, except that thymine is found in DNA. Now, if you take these, so the ribose or the deoxyribose backbone is the same for every nucleotide. What's different is the nitrogenous bases. Okay, so if you take a whole bunch of these nucleotides and put them together so that you have the bases in a certain order, that's how you make a gene. And if you make 2 chains like this with the bases facing inwards, the bases, so here we have, you can see this diagram. The outer part, this part here, is meant to be the ribose or the deoxy. In this is a DNA double helix, so this is meant to be deoxyribose on the outside. So all of this stuff on the outside, this backbone, is meant to be deoxyribose. All of these ring structures on the inside are meant to be the nitrogenous bases. Now notice that they're facing each other, but they're not quite touching, but they are actually bound to each other by hydrogen bonds. Right, so the bases, depending on which bases you have, are attracted to each other by hydrogen bonds, which is what's holding this double helix together like a zipper. Okay, now it just so happens that cytosine will hydrogen bond with guanine, and adenine will hydrogen bond with thymine. Right, so whenever you see an adenine on this side of the double helix, you will always see a thymine on this side of the double helix. Right? If you see guanine here, then you'll see cytosine over here. Right? So that's how the double helix is held together, and that hydrogen bonding between the bases is referred to as complementary base pairing. All right, now forget about the ribose backbone. Just look at the bases. What order did you put the bases together in? Right. So if we abbreviate the bases TACG, and you look at one of the 2 strands, right? So you peel away one of the strands and you look at the other one, and you see that you have bases that go ACTG, ACTG, something like that. That's kind of a code. It's like a written word, isn't it? If the bases are put together in a certain order, that has the ability to carry information. So if you look at every 3 bases, if you take the bases on that DNA strand and you count them 3 at a time, each 3 of those bases is something called a codon. And a codon, one codon, codes for 1 amino acid. Right? So when the cell is trying to figure out which amino acids to put together in what order, it does it by making a piece of DNA and then putting bases together in that order. Putting the bases together in that order, and then you make a temporary copy of that DNA strand. You make an RNA copy that's temporary. You send the RNA copy out to the outer part of the cell, outside the nucleus to the cytoplasm. The That is called a messenger RNA. That messenger RNA will load onto a small thing called a ribosome, and the ribosome will put together the amino acids according to the plan that was present in the gene. So you have these genes in the nucleus that are made of DNA. If you make a temporary RNA copy, that's called transcription. So you make an RNA copy from the DNA, and you have transcribed what's known as messenger RNA. The messenger RNA then goes out into the outside of the cell cytoplasm, and it loads onto an organ called a ribosome. And then the ribosome will read the messenger RNA and then put together amino acids in the order that is specified in the messenger RNA. And that process is referred to as translation. This is known as the genetic code, right? So inside each of these boxes, you have the names of the amino acids. See, there's phenylalanine, there's leucine, there's another leucine. So notice that we have 4 base pairs, but we have 20 different amino acids. So it is possible that more than one codon codes for the same amino acid, so that the genetic code is actually redundant. But so let's look at how to use this table. Okay, so what if we have the codon AUG? By the way, this uses the U's instead of the T's because this is meant to be RNA, not DNA. Right? So you make a copy of the DNA, you make a copy into RNA. The RNA uses U's instead of T's. So let's say that the codon you have is AUG. Okay, so for the first base in the codon, I look down here, and I see the letter A, right? And then the second base, I look up here, and the second base is a U, so it's there, right? And that leads me to this box. It leads me to this box. And then I look and I see AUG stands for methionine, and it's also known as a start codon. So when the ribosome sees the codon AUG, it knows that it's supposed to start building a protein, and the first amino acid that it puts on to the end is methionine. Okay, what if the next codon is CCU? Okay, so the first letter is a C, so I'm going to look here. The second letter is a C, so I'm going to look here. Those two lines converge into this box. And I see that CCU codes for proline. Proline is another amino acid. Right, so you can see how a gene works to code for a protein. Every 3 bases is considered a codon, and every codon codes for one of the 20 amino acids. You transcribe an RNA copy from the DNA template in the nucleus. That's called a messenger RNA. That messenger RNA goes out into the cytoplasm, and it loads onto a small organ called a ribosome. The ribosome reads the messenger RNA, and using the codons puts the amino acids together that are supposed to be put together. So that's known as the genetic code. All right, so to summarize everything we talked about today, we talked about the carbohydrates and how they end in the suffix "-ose", and how you have monosaccharides, disaccharides, and polysaccharides. We talked about how you synthesize them by dehydrating them and break them apart through hydrolysis. And when you put together 2 monosaccharides, the covalent bond that is formed is called a glycosidic bond. And the 2 most important carbohydrates that you need to know are glucose and ribose. Lipids, they are hydrophobic. They're made of aliphatic hydrocarbons. If you put together 3 fatty acids and glycerol, you have triglyceride. That's used for storage fat. We learned about phospholipids, which form the outer cell membrane, the membrane of the cell. We learned about cholesterol, how it's used to make the phospholipid bilayer stronger, and how if you manipulate or you change cholesterol a little bit here and there, you've created steroid hormones. Proteins are made of amino acids. There are 20 different amino acids. Those amino acids are put together in a certain order that is specified by the genes. The genes specify what amino acids they want to be put together through codons, 3 bases at a time. You read that. It's kind of a code that tells you which amino acids to put together. The putting together of the amino acids is referred to as translation, and it's carried out by microscopic organelles called ribosomes. And nucleic acids are made if you put together ribose, either ribose or deoxyribose, and you stick on one of 5 possible nitrogenous bases. ACGT are the 4 that are found in DNA, and then ACGU are found in RNA, so that's the only difference. And then you learned about the genetic code. Now the other thing that's interesting is that ATP is used not just as a nucleic acid for messenger RNA, but ATP is also used as an energy source in the cell. Right? So we learned that the main energy source in the cell is glucose. Glucose is broken down inside organelles in the cell called mitochondria, which we'll learn about later. And the energy that's released from breaking down glucose is used to put together ATP, which is a smaller energy molecule, so it contains less energy. It contains a smaller amount of energy that's more appropriate to the small reactions that are taking place in the cell. Okay, so you might want to try some of these practice questions. What's the difference between saturated versus unsaturated fatty acids? And what's a polyunsaturated fatty acid? What is an essential fatty acid? What is an omega-3 fatty acid? What is an essential amino acid? Right? Sometimes people say that meat or I'll do the last one here. Sometimes people say that meat or fish is a good source of protein, and rice is a good source of carbohydrates. What do you think they mean by this? Well, that's an interesting question, because fish is made, every cell inside a fish is made of carbohydrates, lipids, proteins, and nucleic acids. And every cell that makes up a rice plant is also made of protein, carbohydrate, nucleic acid, and lipid. Right? So if you're eating rice in the hopes of getting nothing but carbohydrates, that would be foolish, because the rice contains a lot of carbohydrates, amylose to be sure, but it also, every cell contains proteins and lipids as well, and nucleic acids. The same is true for fish. Let's say that you're on something called the South Beach diet, which is a diet that people use to try and lose excess weight, where they eat nothing but meat, right? And so they don't eat any, they deliberately avoid eating carbohydrates. So they're deliberately trying to avoid eating carbohydrates, and yet every cell inside a fish contains a certain amount of carbohydrates anyway. So there's no escaping it. Every cell in every living thing contains all 4 of nucleic acids, proteins, lipids, and carbohydrates. It's just that if you're eating the fish, it's mostly muscle. So muscle is made of myosin, a protein called myosin. So yes, if you're eating the flesh of a fish, it is mostly protein. Whereas if you're eating rice, it is mostly carbohydrate, but you still get the other 3. You get the other 3 classes of macromolecules in it as well. Okay, so now proceed to Unit 3, and we'll talk about cell biology. We'll have an introduction to cells. That's still not anatomy, but it's closer. So in the next lecture, we're gonna talk about cell biology, have a brief review of what you find inside a cell. And then we will talk about what happens when you put cells together into tissues. And when we do that, that's the study of histology. That's actually closer to anatomy. So proceed to the next lecture, and we will discuss cell biology. Thank you.